Welcome back to the deep dive. Today, we've got a big one. We're taking that massive, often intimidating textbook, Elsevier's Integrated Review Biochemistry, and we're going to distill it.
Right.
Our mission is to pull out that pure, high-yield knowledge that you can actually use and to cut right through the noise.
And it's really the perfect source for this kind of deep dive. The whole point of the book is that biochemistry is about integration, not just lists of facts.
It's all about connectivity.
Exactly. It's a subject of connections and consequences.
And the source material gives us this brilliant shortcut, a framework that we're going to use today. For every pathway we talk about, we'll hit 5 key points.
Okay.
What are the components? Where are the regulation points? How does it intersect with other pathways? What are its unique features? And, the most important part.
Clinical features. What happens when it all goes wrong?
Precisely.
Using that framework, you really do bypass rote memorization. You move straight into a real functional understanding.
So where do we start?
We have to start at the very foundation. The properties of water and the absolute basics of acid-base chemistry.
Okay, let's do it. Water. It seems so simple, but it's the entire reason biology works.
It is. And its role is all tied to its structure, the way those molecules form hydrogen bonds.
Not just with other things, but with each other, right?
Extensively with each other. And the simple consequence of all that bonding is that water has this, exceptionally high boiling point. If it couldn't do that, it would just be a gas at room temperature like CO2.
So no oceans, no cells, no life.
Imagine trying to do biochemistry in a gas. It's impossible.
Okay, so in that watery environment, we measure acidity with pH.
Yes, and it's so much more than just a number. It's the negative logarithm of the proton concentration.
And that logarithmic part is what trips people up, but it's critical.
It's absolutely vital because it means every single unit change in pH is a 10-fold difference in acidity.
10-fold.
So your blood pH drops from 7.4 to, say, 6.4. Your body is suddenly dealing with 10 times the protons. It's an exponential threat.
And the body needs a way to manage this, right? To fight back against all that metabolic acid.
Exactly. And the mechanism for that is, well, it's described by the Henderson-Hasselbalch equation.
The famous one.
The one everyone remembers. It's essentially pH equals pKa plus the log of the base over the acid. It describes how buffers work.
So these buffers, they're like chemical shock absorbers.
That's a great way to put it. There's systems designed to resist these huge pH swings. Our main one is the carbonic acid conjugate pair. The bicarbonate ion just soaks up any free protons.
Speaking of things being affected by pH, let's talk about proteins, the workhorses of the cell.
And their function is incredibly sensitive to pH. It all comes down to charge.
Where does that charge come from?
It comes directly from the ionizable side chains on certain amino acids. You have aspartate and glutamate, which are acidic, lysine and arginine, which are basic, and then there's histidine.
And histidine is always the special case.
Always. Because its side chain has a pK of around 6.0, which is it's incredibly close to physiological pH.
So it's perfectly positioned to act as a buffer.
An outstanding physiological buffer, especially inside cells or in hemoglobin. It can flip between being neutral and positively charged right around that crucial pH range.
That brings us to another key concept, the isoelectric point, or pI. Why is that number so important?
The pI is the exact pH where the net charge on the entire protein is 0. All the positives and negatives cancel out.
And what's the practical use of knowing that?
Well, at that point, the protein is least soluble, and it won't move in an electric field. So if you're trying to purify it, that's your magic number. It tells you exactly how it will behave.
Let's connect this to a clinical picture. Metabolic alkalosis. What happens when the system gets thrown off, say, by losing too much acid?
If you lose protons, maybe from persistent vomiting, the whole carbonic acid equilibrium shifts. The body tries to compensate.
No.
Well, bicarbonate starts to accumulate in the blood. That's the marker. But the really interesting part is the respiratory system steps in and slows your breathing down.
To hold on to CO2.
To retain CO2, which is an acid, it just shows how fast your lungs are connected to basic blood chemistry.
That's a perfect transition from the chemical environment to the machines that run it. Enzymes. Let's get into the logic of catalysis.
Right. And to study enzymes, we use the Michaelis-Menten model. It's all about measuring the initial reaction speed.
And from that model, we get 2 really important constants. Let's start with Vmax.
Okay, so Vmax is the absolute maximum speed the reaction can go. It's what happens when the enzyme is totally saturated with substrate.
And what does it tell you?
The most important thing to remember is that Vmax is directly proportional to the amount of enzyme you have. Double the enzyme, you double the Vmax. It's a measure of raw capacity.
Okay, so that's capacity. Then we have KMO, the Michaelis constant. People often just say it's affinity, but it's more nuanced.
It is. Thinking of it just as affinity can be a bit of a trap. Technically, it's the substrate concentration you need to reach half of Vmax.
So what's the biological insight there?
A low QRM means the enzyme is very efficient. It doesn't need much substrate to get going at near top speed. Think of it like a scavenger. A high QRM means the enzyme only really kicks into gear when there's a ton of substrate around, kind of like an overflow valve.
That's a much better way to think about it. Now let's talk about stopping enzymes. There's normal regulation, and then there's inhibition by drugs or toxins.
And we can start with reversible inhibition. Two main types. First, competitive.
Competitive sounds straightforward. They compete for the same spot.
Exactly. The inhibitor looks like the substrate and competes for the active site.
So you can overcome it.
Yes, you just flood the system with the real substrate. Because of that, Vmax doesn't change. You can still reach it, but the QRM goes up.
Why does QRM increase?
Because you need way more substrate now to outcompete the inhibitor and get to that half maximal speed.
And the classic clinical example for this
Is methotrexate. It's an anti-cancer drug that looks like folic acid. It competitively inhibits an enzyme called dihydrofolate reductase, which shuts down DNA synthesis in rapidly dividing cancer cells.
Okay, so that's competitive. What about the other type, non-competitive?
This one works completely differently. The inhibitor binds somewhere else on the enzyme, not the active site.
An allosteric site.
Right. The substrate can still bind, so the affinity, the chiral, doesn't change. But once that inhibitor is on, the enzyme is just functionally broken. It's taken out of commission.
So you can't overcome it with more substrate? What happens to the kinetics?
Since you've effectively lowered the concentration of working enzyme, you can never reach the original Vmax. So Vmax goes down, but QBRM stays the same.
This is where it gets so interesting for pharmacology. Let's talk about irreversible inactivation and use aspirin versus ibuprofen.
This is a perfect high-yield comparison. They both inhibit the same enzyme, cyclooxygenase, to stop inflammation.
But in totally different ways.
Totally different. Ibuprofen is reversible. It binds, it does its job, it leaves. The enzyme is fine. Aspirin is a kamikaze.
What do you mean?
Aspirin, the acetyl group, actually gets transferred and forms a permanent covalent bond in the active site. The enzyme is dead. Permanently. The only way the cell can get that function back is to synthesize a whole new enzyme molecule. It's the ultimate form of control.
That idea of control brings us right to the heart of it all. Integrated metabolism. Let's apply our 5-point system, starting with that crucial gateway, the pyruvate dehydrogenase complex, or PDC.
The PDC is the point of no return. It takes pyruvate from glycolysis and commits it to being oxidized in the citric acid cycle by converting it to acetyl-CoA.
Let's start with its unique feature. It's not just one enzyme.
No, it's this incredible piece of molecular machinery, a massive multi-enzyme complex with 3 distinct enzymes all arranged together.
And the unique feature is something called substrate channeling.
Exactly. As the product of the first enzyme is created, it's immediately handed off to the second, then to the third. It never diffuses away.
Like a little assembly line.
A perfect assembly line. It makes the whole process incredibly fast and efficient.
And how is this critical gateway regulated?
The main way is through covalent modification, phosphorylation. When it's phosphorylated, it's off. Dephosphorylated, it's on.
And what signals control that switch?
Energy levels. High levels of ATP or NADH scream, we have enough energy. And that signal leads to phosphorylation, turning the complex off.
Okay, so regulation is clear. What about intersection?
Well, the acetyl-CoA it produces feeds directly into the citric acid cycle. That's its whole job. It's the bridge.
And what happens if this bridge collapses? What's the clinical feature?
The big one is lactic acidosis, an overproduction of lactate.
Which is usually caused by a shift in the cell's redox state, right? Too much NADH.
Correct. Things like hypoxia low oxygen or even excess ethanol consumption cause a massive spike in the NADH to NAD plus ratio. The cell gets desperate to regenerate NAD plus brine, and it does that by converting pyruvate to lactate.
But a specific genetic defect in the PDC itself is even worse.
Devastating. If the PDC is blocked, pyruvate can't get into this K, it piles up, and so does lactate. You get severe neurologic problems.
Why the brain specifically?
Because the brain is an energy hog, and it's almost completely dependent on oxidizing glucose through the CAC. You block that gateway, you starve the brain of its main fuel.
All right, we've covered the chemistry, the enzymes, the energy. Let's pivot now to the blueprint for it all. DNA, organization, fidelity, and repair.
The sheer scale of DNA organization is just It's mind-boggling. You have to compact meters of DNA into a tiny nucleus.
And the first level of that is the nucleosome.
Right. You coil the DNA around these histone proteins. That's the basic unit.
And that structure immediately creates function by separating different regions.
Precisely. You get heterochromatin, which is super condensed and genetically silent, and euchromatin, which is open, accessible, and ready for transcription.
So when this huge genome gets copied, the process is semi-conservative,
Yeah.
but the real magic is the fidelity. How does it stay so accurate?
The primary mechanism is proofreading. Mistakes happen, bases can briefly shift into weird chemical forms called tautomers that cause a mismatch.
But the polymerase catches it.
It does. It's not just a builder, it's an inspector. It feels the distortion in the helix shape caused by the mismatch.
And then what?
It has a backup function, a 3' to 5' exonucleus activity. It's like hitting the backspace key. It stops, backs up one step, cuts out the wrong base, and puts the right one in.
Incredible. Now, there's a unique problem at the very ends of our chromosomes. What solves that?
Telomeres and the enzyme telomerase. The normal DNA polymerase can't quite finish copying the very end of the lagging strand.
So you'd lose a bit of DNA every time the cell divides.
You would. But telomerase comes in. It's a special enzyme that carries its own little RNA template and adds these repetitive DNA sequences, the telomeres, onto the ends.
And this has huge implications for aging.
Huge. Most of our normal body cells don't have active telomerase, so with every division, the telomeres get a little shorter. It's a kind of mitotic clock.
And when the clock runs out?
The cell stops dividing and enters apoptosis, programmed cell death. On the flip side, reactivating telomerase is one of the key things that allows cancer cells to become immortal.
So we have proofreading for accuracy during replication. But what about damage from the outside world, like UV light?
Then you need your repair mechanisms. A really important one is nucleotide excision repair, or NER.
And what does it fix?
It fixes bulky lesions that distort the helix. The classic example is a thymine dimer, where two adjacent thymine bases get fused together by UV light.
How does the repair work?
A set of enzymes recognizes the bulky distortion, cuts the DNA strand on either side of the damage, removes that whole section, and then DNA polymerase fills in the gap.
And the clinical link here is absolutely stark.
It really is. A genetic deficiency in one of the NER enzymes causes xeroderma pigmentosum. These patients can't repair UV damage.
So sun exposure is incredibly dangerous.
Incredibly, they have extreme photosensitivity and an almost 100% chance of developing malignant melanoma, often very early in life. It just shows how vital these systems are every single second.
So let's bring this all together. We've gone from the basic rules of pH and buffers to the kinetics of enzymes, into the metabolic engine room with the PDC, and finally to the blueprint itself, DNA, and how we protect it.
And the thread connecting it all is that integrated approach. You see how a single failure, whether it's in a buffer, an enzyme, or a repair protein, immediately cascades into a real-world clinical problem.
This is all applied knowledge.
Exactly. It links structure to function, to regulation, and ultimately to pathology.
To make sure these connections really stick, we want to leave you with a final thought exercise. It pulls together a few things we talked about.
Okay, so think about this. In untreated type 1 diabetes, you have very high levels of NADH from all the fat being broken down. You also have pyruvate accumulating because the PDC is inhibited.
So given that metabolic state with the PDC blocked and NADH levels through the roof, what is the fate of all that pyruvate? And what major clinical condition results from it?
Think about what the cell needs to do with all that excess NADH. The door to the CAC is closed for pyruvate.
So where else can it go?
Exactly. Go synthesize that final piece of the puzzle.